And I just want to introduce my collaborator here, Danny Haelewaters. Wave your hand. And he's from Belgium. He's been interested in this fascinating group almost 10 years and has done a lot of collecting and is one of the few people in the world who can actually identify species. I've been interested even long before I saw one of them, just from reading about them and their associations with insects and a few other arthropods. And it was through the generosity of John Taylor, actually before the primers were ever published, that I was able to go to his lab and start some work on the group so that I could figure out what was happening. And so they have been called algae, red algae, and this happened way into the last century, the end of the last century, when Cavalier Smith called them zygomycetes. Many people along the way, even well into the 20th century, called them red algae. And they were described as worms, the second genus ever described. So it's been something of a cliche to call them enigmatic. And so I hope to show you today that they're really not enigmatic. We know what they are, and I hope you'll agree in our hypotheses of how they got that way. So these are all arthropod associated, the whole class. Some of them, the ones that we know best, are all parasites of insects. We also have a number that are phoretic on insects, and I'll talk more about those. Those are the ones that David has worked on. They're obligate biotrophs, which is sort of repetitious there. They have a thallus, which is mycelial, or in the case of the ones that we know best, they're actually cellular. And I hope also that you'll understand what we mean by a Transchell's law for Laboulbeniomycetes at the end when I talk about that. Now, here's Danny's latest tree. This has all of the lobules that have ever been sequenced. They're very difficult to sequence. Most of them don't grow in culture, especially the ones in the Laboulbeniomycetes. Whistler cultured some, although they were two-celled plus a few conidia, the male thalli of a few species. And then the Pyxidiophorales are very difficult to culture. Most of them are mycoparasites. Walter Gams has been able to bait some of them with sclerotia, and he had some that grow in culture without having a host. So those are very useful, and several of them are here at CBS. Now, I've put a little bit about the families here, and Danny has counted up all of the numbers here on the right-hand side. The top group of numbers goes with the Laboulbeniomycetes, and you can see that most of the genera were described by one person, Roland Thaxter, and these are at the Farlow Herbarium. Then there have been some others added, and not too many, but there has been more recent work with Danny's collaboration with Walter Rossi and some other people. So there are new species being described. Now, they're an estimated number, 15,000 to 75,000. I'm not sure the basis of this, but anyway, it is thought that there are a number of them that are out there that we don't know anything about yet. And so they've been grouped into four families, and then there are two other genera that I've put here. One, Laboulbeniomycetes, that I'll mention, is in Mycobank, listed in the unassigned to a family. And then the Pyxidiophorales, there's one family, Pyxidiophora, with a few genera, and these are the genera that are listed in Mycobank, and I'll talk more about them later. So here's what we think of most of the time when we talk about Laboulbeniomycetes. These are the ones that are associated with insects as obligate parasites. The CETA is shown at the yellow arrow. It's sort of a yellowish color, And as an extension, the two blue arrows there are pointing to what are Laboulbeniomycetes, and this is a passalid beetle. And so they're very difficult to see, and many entomologists work with some of these fungi and never actually see them. I mean, they work with the insects and don't see them. On the right, you have thalli of another group of these that are on Nectarobiid bat fly. Now, Alex Weir, for his PhD dissertation, did a nice study and did some beautiful micrographs. You can see there on the left, number 1, this is a 2-celled spore. And when you do electron micrographs, scanning electron micrographs, you don't see the cell divisions very well. But these are 2-celled, and the white arrows point to the attachment site of these spores. If you look at number 2, that is a scar that was made by a labule on the insect cuticle. And then 3 shows the development from the spore. The black arrow indicates the septum of the 2-celled spore when it was still a spore. And the red arrow points to a spike that was at the tip of the spore. And you can see it's become attached to the cuticle of this bat fly in this case. And number 4 just shows one of the species that is, it's got a separate male and female phallus. Now, this is a tree then that shows that along with these Laboulbeniomycetes that are not really very well supported. Let's see if I can get this to work without. You can see that there's no support here for the 2 groups that are well supported. And Herpomyces is a genus that's thought to have been diverged very early in the evolution of Laboulbeniomycetes along with roaches. But here it was sort of a surprise. This is Laboulbeniomycetes, which has also come in here. And this is a very small phallus, and I'll show you that next. And here you can see this fungus. It's restricted to termites. And there are actually 2 genera here, both described by Faxed. On the left is Coriomycetopsis, which has not been sequenced at all. And you can see these were drawn by Faxed. And on the right is the Laboulbeniomycetes, and also a photograph from material from Louisiana that was on the tip of the antenna. This is the tip ball of the antenna of a termite. So they're very, very tiny, about 200 micrometers. And in a study I did a long time ago with Kimbrough in Florida, we had assumed that perhaps, based on some electron microscopy, that these were ascospores that had developed. Now, Daniel Hink, who was an undergraduate student in the lab, was actually the person who would come in late at night and take these and have a huge number of the thalli taken from termites, and he was able to get some sequence. And I should point out that there's not much sequence for any of these. Only ribosomal genes have been sequenced for any of these Laboulbeniomycetes. Now, also, something that may add to the large numbers, Danny has recently found that of one species, supposedly one species of a labule of Hesperomyces virescens, he actually can find fairly good evidence here that these different strains that were removed from ladybird beetles, one here from Utrecht, which I thought was interesting, and these are all very similar, identical, right? The sequences are. And so it seems that if you look at the different host beetles of this, that there actually is perhaps some cryptic speciation. So this is something pretty new. It might even add to numbers of species. It's been a big question. If you look across the world, some of these things that have been described seem to have very, very broad distributions, which doesn't seem right. Okay, so the Laboulbeniomycetes, then, to summarize this group, they're biotrophic parasites on arthropods. They have two-celled ascospores that develop into a thallus that may have several cells, several thousand cells. They have determinate growth, so that each species is characterized by pretty much a common number of cells. And this is something that was used by Isabel Tavares to arrange families in a different way from the traditional way. They've also lost the ability to germinate by a germ tube, therefore there's no mycelium. And this is a character that has caused people to wonder what these were and describe them as algae or worms. And also, unusual for a group of fungi, they've lost not the ability to reproduce sexually, but they've lost the conidial state, we think, based on the phylogeny. Now, one day I had been working with some people at the Forest Service lab in Pineville, Louisiana, and that's where our mental hospital is. So it's kind of strange when you talk about it. But I got this phone call from Thelma Perry. She worked with bark beetles and the fungi and bark beetle galleries. Thelma was an amazing person. She was someone who could look at fungi and kind of have some idea what they were. She was completely self-taught. And later she did have a course, a mail order course taught by Bryce Kendrick. But anyway, Thelma called and said that she found the parathecium that produced Faxed Areola. Now, this was really amazing because Faxed Areola was thought to be a separate little organism. And you can see there, it's stuck on a mite. It's a bit hard to see. The mite's been cleared and it's also been heated. And all of these black spots represent the attachment part of the Faxed Areola. And there's Thelma working away. So your question might be, what is Faxed Areola? And so all of these on the right, in the square on the rectangle on the right, these are Faxed Areolas. On the left, we have what's been called Areola that's different because it's a little bit smaller in size, but it's also got this pore in it. So these two fungi were named by Faxter. Spagazzini also studied some. And they were thought to be separate organisms. But what Thelma had found was that the neck of the parathecium, which is shown here, and this one's actually sticking out of dung. It's not one that was in with the bark beetles. But the spores are here, and they're very long spores. And the tip is the darkened foot part. Now, these are unusual in a number of ways because almost all of them produce three-spored asci, which is something that's not very common. And what's done is one of the nuclei is actually enclosed in an autophagic vesicle and is eaten away. So it's a very interesting fungus for a number of reasons. Now, it also produces at one end, while it's attached to a mite, it's producing conidia, and so you can see them there. So when the mite, which is sporadic on a beetle, arrives at the next site where it's going to develop, and perhaps a pine tree, you find that the spores are ready to go, and they're right there. So, ascospores mature slowly, and it's unclear to me when you measure the ascospore and decide that it is mature. Here is an ascospore that was in the parathecium, and these, by the way, are not dispersed they're not shut off. They ooze out, and this is the tip part that comes out first. And so you have all of these at different stages of maturity, and some of them are attached to mites. Sometimes in certain isolates, you find that the spore may be darkened. It may have this darkened attachment region before it emerges from the parathecium. So let me go back. No, this is right. Okay, so this is Endospirella, which is a similar kind of thing described by Faxed. And there are Pyxidiophores that have spores that are 500 and 600 micrometers long, and it's quite possible that this may be derived from that kind of thing. And in addition to having these dispersal anamorphs, as we call them, there are also hyphal anamorphs. And Keith Seifert has worked on the one on the left, the gliocephalus, and then on the right you can see some of the conidial development in some of the ones that David Malik and I studied on moose dung. And so if you look at all the names that are in Mycobank, and this was taken directly from Mycobank, and I've organized them into the different types. The parathecial teleomorph names are shown in the top panel. The hyphal anamorphs are in that next panel, and then at the bottom are what we're calling the dispersal anamorphs. And most of these names here are synonyms of Pyxidiophora, and I think we're going to be able to add one more, which is the Mycorhynchus. Actually, I've looked at the type, and there is a darkened region on one of the spores, one out of about maybe 60 or 70. So they develop very slowly over time, and so if you don't catch them when they're fairly mature, you won't see this. Also, there's another species that may be one that's immature because it had it was queistothecial as described by Malik and Kane, but that probably may not be one that was even a mature fruiting body. Now, if you look in GenBank, Kevin, there's one or two of yours here that may be actually Pyxidiophora anamorphs because they have been described as chaleras. And in fact, I was once told by the USDA in the United States that I could bring in Pyxidiophora, but not its parasitic cholera anamorph. So this kind of thing can sometimes be a problem. If you've got an anamorph name that's the same as a parasite name. Also, there's some uncultured fungi. I think John talked about the rind of cheeses. And so there's some of these that might be Pyxidiophores, although it's hard to tell because of the gene regions that have been sequenced. And there are a few others. So they may be more hidden away in GenBank, but there's certainly more out if you know how to look for them. And you have to look very early in the succession for most of them. Now, so here, this is a summary then we know of two sequences or two organisms that are sequenced in GenBank, Keith's Geocephalus and Gliocephalus in the several Pyxidiophores. Now, another problem that's caused confusion with Pyxidiophora is where does it grow? And if you go back to the first one, the type, which was found on Asterophora, the parasitic mushroom shown here on a mushroom host. One of these was the first thing that I saw that made me think they might be insect dispersed. And so that has turned out to be true, and it makes me wonder about the Asterophora, if that might be insect dispersed as well. And if you look at the Encyclopedia of Life, you find that Pyxidiophora interacts with many, many things. I don't know, I've never seen a powdery piggyback, but I did look it up, and it's some kind of plant. But anyway, foxes, jackals, horses, zebras. And so I think this was a way I don't often think of fungus as interacting with all these organisms. But of course, it is found on these substrates. And if we look at substrates where I have actually collected, you see John Webster's foot there in a Dartmoor stream. And he and David described the species that was found on this wood. He knew exactly what to do. You incubated at low temperatures, 15 centigrade or something. And so what he did was keep it. And I think it was about 3 weeks. And then he sent me the anamorph of the Pyxidioptera. And the thing about it is that most of the Pyxidioperas are very quick to come up in the succession. And so this is a really remarkable one that comes up much, much later, or develops much later. At the time that David Malik and I were messing around in this brown algae, we did not know that it's actually a host for Pyxidioptera. And when I say host, that's probably inaccurate. And these are places where we found Pyxidioperas and isolated them. Because what these truly are, as Keith knows, they are mycoparasites. And in the only 2 cases I know of where these have been grown in culture and they're not mycoparasites are the 2 strains at CBS that Walter had isolated. So if you look at the Pyxidioptera ascospores, the long darkening ones with the dark attachment foot, you can put them onto shards of glass, and then the glass can be laid down so that you've got the cover slip over the spores that are developing, and you can watch what happens. And what happened here was that these tips, there were yeast cells that developed from the Areola, and then they sat there until a host came nearby. And this fungal host was in the substrate. And so this explains why there's so many substrates that this fungus interacts with. It's because it's really the fungus on the substrates. So there is a common entity there. And this is from Keith's paper showing contact mycoparasitism in a really nice way, and these are beautiful micrographs. So the Gliocystis, or the Pyxidioptera anamorph, can be seen here, this one, and here is the host, and here is this other one shown with TEM. So if we review the information about the Pyxidioptera, they're biotrophic, but their hosts are not arthropods, as are the other Laboulbeniomycetes, the other order, but they're fungi. The teleomorphs are unusual because they have a single wall layer, a single cell wall layer, and there's only one other fungus that I know of, one other parathecial ascomycete that I know of that is this way. And that's something called Cathistes, which is somewhere near here, but it's certainly not a sister taxon. We have also 2-celled ascospores that look exactly like the Laboulbeniomycetes ascospores, and we've called them dispersal anamorphs, which would make a 3rd morph in this life cycle. There's germination of the yeast that's produced by the Faxed Areola morph, and these are able to germinate by a germ tube. And of course, they're the mycelial anamorphs that are Cholera-like or Gabernodia-like, as they've been described. And so if you look at all this, it gives you some ideas by looking at the life cycle, and this is going way back again. Not only do we use morphology, but we use life cycles to try to figure out fungi. But it's it gives us some ideas, and these are some characters that we've put here that indicate where we think certain things have happened. We're hampered because there is this very, very long branch, and I didn't point it out before, but the Laboulbeniomycetes are the sister to all of the Pazizomycetes. And so here, there's no forcible spore discharge in an ancestor, probably, sticky conidia perhaps. They're biotrophic, and there's an elaborate ascospore foot that has developed here in these two groups. And these are things that had led Cavalier Smith to put them into zygomycetes that attach to things. Also, we find that here with the Laboulbeniomycetes, they're restricted to arthropod host. You can kind of think of them as being microcyclic. They've lost the conidial state, and perhaps that conidial state has been transferred onto the thallus so that it is it has a spermatial function, and also this determinate growth. And so these characters that have asterisks are things that are unique to these groups. So what do we need? We need more people who know what these are. One of the worst things is, and this has been pointed out, I think, by Kevin, we need people to go out and find them because they don't grow in culture well. And certainly no one really has grown any of the Laboulbeniales. And so this is a real need, people who can identify them and find them. And we also need more genes and more taxa, which will come if we have someone who can go out and get the fungi. But that's extremely important. And I should say that the people who work with Labuls, including me on occasion, we need to express these parts, the structures, in a way that other people can understand them, because the thallus of this Laboulbeniales order is very, very different from other fungi, and so it's been difficult for people who aren't specialists to be able to understand them. And so we've got some thank yous here. Danny got here because of Harvard money, and the National Science Foundation has funded me for several grants to work on these fungi in the past. We also had an unpublished sequence from Hungary. And then I want to do a change of address. Oh, they're Labuls. Okay, so when I still use an LSU email. I always will. They've said I can keep it till I die. And so that you can always find me there. But I have moved. I live now in Columbia, South Carolina. And it's a place that is very southern U.S. But what's important to know is that Dreher High School, which is 3 blocks from my house, is the place where Carey Mullis went to high school. And in his Nobel address, he made the comment that at Dreher High School he was allowed free access, unsupervised, to the chemistry lab. And so living 3 blocks from this school, I do hope they've changed the rules. But anyway, so I am in South Carolina, good collecting, under-collected. So I hope some of you will visit anytime you want. Thank you.